Hello friends, welcome to our channel Medicine and Health Initiative Center. Today's topic is complement activation system. We are going to look what is complement, the function of complement system in the clearing of pathogen within the body. So, by definition, you are going to see complement system. Once we say complement system, it is a system of proteins made within the body, especially in the liver as human organ. Those complement comes to complement the immune system. And as I have said, they are proteins. They are proteins which interact with the pathogen. After interaction with pathogen, actually it marks the pathogen for being destructed via phagocytosis. So the main function of this complement system, it involves phagocytosis, opsonization, and to induce inflammation. Phagocytosis, opsonization, and induce inflammation. So this complement system, it involves series of pathways. It's about 3 pathways, which are classical pathway, focus on alternative pathway, and mana binding pathway. Focus on classical pathway as the first step, and we are going to look the mechanism. In the classical pathway, we are going to see different proteins which involved in this classical pathway. But first things that we have to know, it is initiated by antigen-antibody complex. Once antigen interact with antibody, there will be a formation of complex. As long as this complex is formed, this process actually starts. Within the circulation, as those complement or protein produced, the goes through circulation, have different protein, they are produced in inactive form, so they need to be activated so that this process should be progressed. We have those protein include C1, which is made of C1Q, C1R, and C1S. So we are going to start this process, how it takes place. As this complex is formed, the C1Q will come to bind to this complex to initiate the activation. After binding, this C1R, it will activate C1S. The activated C1S this C1s, after being activated, it will act like a protease. It will cleave some complement within the body, including C4 and C2. This C, it stands for complement. It means that we have C4 and C2 complement. We are going to see how the mechanism progress. C4 and C2 will be cleaved by C1 as they have been activated. This is C4, it will be cleaved into C4b and C2, C4a. And C2 will be cleaved into C2a and C2b. And these are the product of C2 and C2a degradation. This is C4b and C2b will form a complex which is stable. It is called 6-3 convertase. This 6-3 convertase, it will come to activate C3. This is a complement 3 which is in circulation and is in inactive form. It needs to be activated. The activated C3 will give us C3b and C3a. This C3a, it will involve in inflammation, to induce inflammation. And this C3b, it will involve in binding to the pathogens, and also it will activate C5. This C3b, it will activate C5. This activated C5 will give us C5a and C5b. The C5b produced, it will activate other inactivated proteins via circulation, which it will activate C6, C7, C8. After activating C6, C7, and C8, there will be formation of what we call MAP. MAP, which stands for membrane attack complex. This complex formed, it will be formed on the surface of on the cell membrane or the surface of the pathogen. It can be bacteria, it can be fungi, depending on the kind of pathogen that the body wants to clear within the body. And this MAP formed, it will create the pores. It will create the pores on the surface of pathogen. After creating the pores, it will lead to the influx movement, where water and sodium, it will winter within the pathogen, and it will lead to cell which will somehow become big, and it will lead to cell death by lysis. So we are going to look the function of those activated complement, so that and how it involves in the clearing of the pathogen within the body. We have seen that C3A, C4A, C5A. This activated complement C3A, C4A, and C5A, they will involve in inflammation. They will induce what we call inflammation. they will C3A, C4A, C5A, they will involve in inflammation. So those inflammation, they will come once it will bind on it will induce cellular permeability by increasing the leakage on the endothelial cells. And we have this cell. So to increase the leakage, you just get a cell like this. On this part, to increase the leakage. After increasing the leakage, the macrophage and other phagocytes will become on the site of infection to clear the pathogen. That's what we call anaphylotoxin. They will act as anaphylotoxin by the process of anaphylaxis. As long as those macrophage and the phagocyte are produced, actually they will involve in the clearing of the pathogen on the site of infection. Another thing that you are going to see, the function of C4B, C3B, and C5B. These are complement which involves in what we call opsonization. Actually, they act as like opsonin. Opsonin, these are the protein which induce opsonization. By definition, opsonization is the process by which this complement will opsonize, it will mark the pathogen, it will put like a marker, so that the marked pathogen will be ready for destruction by phagocytosis. So it's all about the classical pathway and how it how it involves in the clearance of the pathogen, leading to increasing the immunity. And the things that you have to know about this complement system activation, it is a part of innate immunity, because actually the body is the one which evolves to clear the pathogen. So thank you for watching this video.